Welcome back. This is our second video on mitosis and meiosis, and here we're going to start taking a look at mitotic cell division. So we'll deal with mitotic cell division and later we'll do meiotic cell division. So let's get straight into it, and let's start off by thinking about the functions of mitotic cell division. So mitotic cell division is when one somatic cell becomes two somatic cells. So a somatic cell is any cell in your body that doesn't give rise to sperm or eggs. So those would be called the sex cells. So we're talking about somatic cells, all the other cells basically, skin cells, muscle cells, those kinds of things. So that kind of leads us to our first function really. During embryogenesis, you have to produce a certain number of cells which can then differentiate to become all the different types of tissue. So one of the main functions of mitotic cell division is to produce enough cells so you can form an adult organism. And then once you get an adult organism, obviously you lose cells on a daily basis. Just skin right now is just cells are just sloughing off. And so you have to replace those. So that's the other function. Another function of mitotic cell division is to replace cells that you lose just as a course of kind of the normal business of being alive. And so you've got to think about your skin, you could think about cells that are lost from your gastrointestinal tract when material passes through and then out of your body. If you sneeze, then obviously you're going to lose some cells when you do that kind of thing. So maintenance and replacement of existing cells is one of the functions of mitotic cell division. Also, there are certain protozoa which actually use mitotic cell division as a form of replication, and so that's important for them, obviously. And then when you have certain physiological events, like you get a wound or something like that, you need to produce new cells so that you can replace the ones that are being lost. So we've got maintenance just as a normal part of living, but then we've got replacement when we have injury and that kind of thing. So let's now kind of take a little bit of a look at the mechanism or the process of mitotic cell division. So I'm going to talk about what's called the mitotic cell cycle. And we break this generally into 4 stages. Let me get a pen so I can mark this up a little bit rather than just talking over it. So when we talk about the mitotic cell division cycle, we often start talking about it at a stage called G1. This means gap 1. So this is just a period of time in the cell when the cell is growing and it's getting ready basically to go through the process of cell division. So that's G1, and this is kind of a cycle, so it's going to be kind of going in this direction. And it's obviously a cycle, so you could draw it the other way around if you liked, but we're going to go in that direction. So following G1 is S phase, and S phase we're going to talk about in the next slide because it's important, because that's when DNA replication occurs. So if you think about it, if we're going to take one cell and divide it to make 2 cells, then we have to replicate all of the DNA so that both new cells, they're called the daughter cells, they both get an exact complement of the chromosomes we talked about last time. So that happens during S phase, and then following S phase is a period of growth called G2, gap 2. And then this precedes a series of events called M phase. So M phase is all of these events here. And so we will then take a look at the events of M phase in a little more detail a little bit later. So M phase is broken down into 2 main events. The first event is called mitosis, and mitosis is where you segregate the chromosomes. So you get the chromosomes and you separate them apart, and then you divide the cell. And so that's called mitosis, and it's broken down into usually 5 or 6 stages: prophase, prometaphase, metaphase, anaphase, and telophase. So we'll talk about those 5 stages in detail. Then following the segregation of DNA in the cell divides, and so we start with one cell like this. It basically pinches into 2 cells. And so that process is called cytokinesis. Now, this is really important because this is about the language we use to describe this process. So M phase is mitosis plus cytokinesis. Now, if you're just talking about mitosis, you're not talking about cytokinesis. But if you talk about M phase, then you are talking about mitosis plus cytokinesis. So make sure you get this language correct. So the cycle just keeps going. One cell can divide into 2, 2 into 4, 4 into 8, 8 into 16, that kind of thing. So let's take a little bit more of a closer look at S phase. So what happens in S phase is the chromosomes are replicated. Remember, if you're going to divide one cell into 2 cells, then you must replicate each chromosome so that you have two copies of that, and then you can put 1 copy in each of the new cells. So here's a chromosome. Let's grab that pen again. So here's a chromosome. It doesn't matter what chromosome that is. That could be chromosome 1 from mom, and then 1 from dad would be somewhere else. And so when we replicate this molecule, we end up with this structure here. Now this is still a chromosome, but it's now called a pair of sister chromatids. And so let me just mark on here. Here's one sister chromatid, and here is the product of replicating the DNA, and we get a pair of sister chromatids. Now, remember, this is not a homologous chromosome. It's got a homologue somewhere. If this was chromosome 1, and let's say that's chromosome 1 from mom, then there'll be chromosome 1 from dad somewhere else. But here we're talking about a pair of sister chromatids. And if we look at them in a little bit more detail, here is one sister chromatid on the left, and then here is another sister chromatid. So what we're going to do in mitosis, when we look at that, when we look at DNA segregation, is we're going to take one of these sister chromatids and we're going to pull it in this direction to one end of the cell, and then we'll take this one and pull it in the other direction to the other end of the cell, and we're just going to divide the cell down the middle. So each cell gets one of the sister chromatids, which then goes back to being called a chromosome. So that's DNA replication. We're going to talk, we already talked about the mechanism of DNA replication, so we don't need to get into that. So we aren't going to talk about the gap phases. We're going to talk exclusively about mitosis, which I'm looking at now here on this slide, and we just talked about DNA replication in S phase. So the rest of what we talk about is going to be about mitosis. So those are the events by which DNA is segregated. So there's a structure which is critical for doing DNA segregation in the cell, and that's called a spindle. And so this cell here is about to divide. It started the division process, I guess, actually, and it's formed this structure called the mitotic spindle. And the mitotic spindle is made from all of these microtubules, and they radiate out into the middle of the cell. And there are some which kind of go at the back here a little bit, but we're not going to worry about those. So those are those little ones there. We're not worried about those. Those are called astral microtubules. We're worried about these big long ones which kind of come down in the middle of the cell. And they form this structure called the spindle. Here's what the spindle does, is it grabs hold of the pairs of sister chromatids that have lined up in the middle of the cell. So at this point in mitosis, all the chromosomes have lined up in the middle of the cell along an imaginary structure here called the metaphase plate, and we're going to talk about this in a lot more detail in a moment. And so what happens is that each of these spindle fibers, grabs hold of one sister chromatid, and so that one is grabbed there. This microtubule coming down from the top has grabbed this sister chromatid, and when they've been grabbed by the microtubules coming from each end of the cell, the microtubules then basically just pull them apart, and so the chromosomes are going to be dragged in opposite directions, or the sister chromatids will be dragged in opposite direction. One's going to go up and one's going to go down. Obviously, don't forget, I'm just saying up and down because that's how this image is oriented on the cell. So all we do is we go through S phase and we take one chromosome and we replicate it and make a pair of sisters, and then in mitosis we take those sister chromatids and the microtubules grab hold of them, and they come from each end of the cell and they get pulled apart, and that's called DNA segregation. That's what mitosis does. Then we'd have cytokinesis, and we're going to divide that cell right down the middle. Oh, that's weird. Right down the middle of the metaphase plate to give us 2 new daughter cells. So that's a big kind of high-level overview of mitotic cell division. And what we're going to do next is take a more detailed look at the events of mitosis. So we'll talk about prophase, prometaphase, metaphase, anaphase, telophase, and then we'll add on cytokinesis at the end. So I'll be back soon, and I'll see you then.